Good day, everybody, and thank you for joining and welcome to this webinar, which is focused on identifying the right hit finding approach for you. My name is Chris Rutten, and I'm a program manager within the Research Service Division of Wuxi AppTec, with a background in neurobiology, pharmacology, and drug discovery. Before joining Wuxi, I've worked at Grünenthal in Germany as a preclinical pharmacologist and project leader in the field of pain and inflammation for over 10 years. I obtained a PhD in neuroscience from Maastricht University in the Netherlands. I will be moderating this session today, so let's get started. We are increasingly targeting therapeutic modes of intervention that were not previously feasible, and we are working with targets we had not previously identified or considered possible as points of intervention. This progression has been enabled by a combination of development of novel chemical modalities and advances in the techniques available to us to identify appropriate chemical matter. During this workshop, resident experts from Wuxi AppTec will discuss a range of different hit-finding technologies which can be applied either individually or in parallel depending on the target. This will facilitate finding tractable hit matter with the highest chance of being optimized into a drug. Topics that will be covered in detail include high-throughput screening by Dr. Jigang Bang, Affinity Selection Mass Spectrometry, ASMS, by Dr. Penny Jia, DNA-encoded library screening, DEL, by Dr. Alex Satz, fragment-based screening by Dr. Saskia Villinger, and finally, virtual screening by Dr. Dave Madge. Across all these platforms, the process of identifying the most appropriate hit-finding methodology is enabled by experience sharing between our seasoned drug hunters and the client's expertise. This results in carefully constructed proposals with comprehensive, fully costed hit-finding scenarios. There will also be a time available during this workshop to ask the expert in the form of either a panel discussion or directed questions after this session. Wuxi AppTec provides a broad portfolio of R&D and manufacturing services that enable companies in the pharmaceutical, biotech, and medical device industries worldwide to advance discoveries and deliver groundbreaking treatments to patients. As an innovation-driven and customer-focused company, our open access capabilities and technology platforms deliver quality service and help customers and partners achieve their goals more quickly by lowering the cost of research and development and shortening the time required to discover and produce new medicines. With industry-leading capabilities, such as R&D and manufacturing for small molecule drugs, cell and gene therapies, and testing for medical devices, Wuxi AppTec's open access platform is enabling more than 4,400 collaborators from over 30 countries to improve the health of those in need and to realize our vision that every drug can be made and every disease can be treated. Wuxi AppTec's Research Service Division supports clients in the discovery and development of future therapeutics. Through comprehensive discovery and technology platforms, peer-recognized scientific leadership, and expertise in oncology, immunology, infectious diseases, neurosciences, fibrosis, cardiovascular and metabolic disease, RSD works collaboratively to rapidly advance programs from target through to preclinical selection and beyond. RSD's discovery platform encompasses biology, chemistry, structural biology, biophysics, and e-ADME-PK. This integration of capabilities allows a collaborative environment, enabling enhanced efficiency and productivity. Wuxi AppTec's Research Service Division is trusted by over 3,000 international clients, ranging from pharma and biotech to academia and non-profit institutions. Here you can see some examples of awards and contributions to novel FDA drugs that Wuxi has contributed to. In Europe, we have a dedicated project management team created to serve clients requiring support across multiple technical disciplines with design, coordination, data interpretation, and medicinal chemistry optimization components, as well as biological support. We have a global team with strong EU and US presence for technical direction and client interaction, supported by the China team for local coordination and communication. The team consists of experienced project managers, medicinal chemists, discovery biologists, bioinformaticians, computational chemists, and DMPK experts. The team is led by Dr. Dave Match. He is Vice President of the Research Services Division of Wuxi AppTec, and he will give a talk later in this session. Additionally, EU-based DMPK and toxicology experts are available to provide input on appropriate screening cascades. Our mission is delivering comprehensive end-to-end solutions for creating, identifying, and supporting preclinical candidates from discovery all the way through development with our integrated chemistry and biology research platform. And today's workshop will be focused on hit finding. It is our mission to deliver comprehensive end-to-end solutions for creating, identifying, and supporting preclinical candidates from discovery to development with our integrated chemistry and biology research platform. We can offer comprehensive platforms from target ID to IND. However, today's workshop will be focused on hit finding technologies. We have a very packed session with 5 expert speakers that will each share their expertise and demonstrate our capabilities in the following technologies. High-throughput screening, in which we can screen activity of 300,000 compounds against your targets in a 384 or 1536 screening format. ASMS, in which we can screen binding of over 300,000 compounds on SEC plate LCMS and 2D HPLC-MS, warranting high sensitivity, resolution, and accuracy, and a very high throughput. Next, we will talk about DNA encoding library screening, DEL, capable of screening over 80 billion compounds, generating nanomolar binding hits over several target classes. Then we will talk about fragment-based screening, assessing a 3,500 member novel fragment library. Fragments are screened by SPR, NMR, DSF, or X-ray, leading to biophysical hit confirmation and validation with fragment complex structures, biophysics, and biochemistry. And finally, virtual screening, which is hit identification with molecular docking and machine learning, using in silico virtual library screening with on-the-fly enumeration and make-on-demand libraries. So without further ado, I would like to introduce our first speaker of today, Dr. Xigang Peng, who will share his experience about high-throughput screening. Xigang has over 14 years of experience in high-throughput screening, SAR, and assay development. He has a degree in molecular biology and has worked at several CROs before joining Wuxi AppTec 9 years ago. He is currently the Executive Director of the High-Throughput Screening Department of Wuxi AppTec HDB. Xigang, the floor is yours.
Hello, everyone. This is Xigang Peng from Wuxi AppTec Research Service Division, HD Bioscience Department. I'm currently in charge of the high-throughput screening service platform in HD Bioscience. So today, I'm glad to present the Wuxi HDB capability in the HTS area, and also glad to show how we could do to support your R&D activity. For me, I joined HDB in 2006, and in the last 15 years, I mainly focus on the HTS area. So in vitro pharmacology is a key platform in HD Bioscience. So we have developed different assay format and the technology to support the in vitro pharmacology service. So we have full experience on the biochemical assay and the radiometric assay and cell-based assay and high content assay for our clients HTS effort. And besides of this traditional HTS assay and the technology, we also have the biophysical team to support the hit confirmation and fragment-based screening. Also, we use the mass spec as a readout for the label-free screening by using the rapid fire from Agilent and the data from the AppTec. So all these technology and the platform enable us to support the screening target at routine, majority of the routine target, like a GPCR, enzyme, protein-protein interaction target, ion channel and transporter, and nuclear hormone receptor, and the RNA target. In the right of the slide is the key instrument that we used to support the HTS service in HD Bioscience. So you will find the most of the routine HTS reader, and we used to support the HTS and the in vitro pharmacology, and also the different kind of the liquid handler to support the liquid automation in the HTS. So majority of the key reader and the key liquid handler, we have more than one set to ensure different instruments can be back up each other during the HTS screening. So here is some of the track record for the HTS service in HD Bioscience. So we first start HTS in 2009, and so you will see the data point delivered yearly slowly grow. And in 2012, HTB start as a primary HTS service center for two major farms. So we will see a big boost of the data point delivery annually into about 5 million data points per year. And in 2016, majority of the assay, HTS assay, were run in 1536, so enable us to screening up to millions compounds in a campaign. So in last 5 years, we yearly deliver about more than 20 million data points per year to support our clients' screening effort. So this slide is a further summary of the different target classes in the screening campaign in last 11 years. So you will see the HTS campaign we done spread in the different target classes and the different assay format. And HDB will continually deliver the higher hit confirmation rate and the low plate failure rate in the industry average standard. Okay. So information management is of critical importance in HTS screening. So information tracking from the very beginning, from the compound plate into the assay plate and into the data, is the critical for the HTS information management. So in HDB, we have proprietary developed the compound and plate management system to enable us to track the compound information from the very beginning, from the source well to the assay well. And the screening data, HTS screening data, will be analyzed by the most commonly used industry data analysis software like gene data and activity-based from IDBS. And electronic notebook and all the information will be captured in the ELN provided by the ArraySpan or protein era. And this data and the electronic notebook can be easily shared with our clients. So currently, we using the fully automation screening system internally to support the high-throughput screening up to 2,000 plates in a campaign. So this automation hardware is from the PAA automation, which enable us to run HTS almost 24 hours per day and 5 days per week. And this will help increase a lot of our efficiency and help to reduce the human error in HTS screening. So HDB, we not only screening our client library, but we also provide our own library for clients. So current Wuxi AppTec HDB HTS library collection is about 306,000 compounds. We filter the compound from using the different criteria to make sure our library represent a great diversity of the structure. So about half of the library is from commercial available compound library company. And we also have some of the focus library, like protein interaction, nature product like library, and the bioactive library. So all these library were in pre-made 384 and the 1536 well format. So enable us to provide the HTS screening in a timely manner. So the last slide is also more detailed represent of the HDB's Wuxi AppTec compound collection. From the commercial source compound we purchased from Lab Chemical, Maybridge, and Cambridge, and together with some of the Wuxi AppTec chemistry design compound and the biotech collection, enable us to provide a good compound collection source for our clients. So that's my last slide, and I'm glad to take up any of the questions and look forward to have a chance to work with you all. Thank you.
Thanks for a great talk, Sigang. Our next speaker is Dr. Penny Jia, who will talk about affinity selection, mass spec screening. Penny is the ASMS project leader at Wuxi AppTec. She's responsible for data processing and data analysis in ASMS projects. Penny joined Wuxi AppTec in 2019, and before that, she received a master's degree in chemistry from the City University of Hong Kong. Penny, please, the floor is yours.
Hi, I am Penny Jia. I am the leading analyst in ASMS project in core analysis service. And I'm really honored to be invited to this workshop talking about our ASMS platform at Wuxi. I'm going to talk about the principles, our workflow at Wuxi, the advantage of using ASMS, introducing our ASMS library, the capability and advantages of doing ASMS at Wuxi. Oops. Affinity selection mass spectrometry, which is in short ASMS, is a high-throughput screening technology for drug discovery. It could rapidly screen a large collection of chemical compounds in order to identify ligands for specific biomolecular targets. ASMS definitely gain more attention these days, and unlike other technology, the functional effects of this specific biomolecular target is not important in ASMS. In this case, novel ligands could be identified regardless of its binding site. So, how does it work? The small molecule and target are first incubated together in solution. Then a separation is conducted and only leaving us the target and its binders. On top of that, distillation step is carried out and the binder will be analyzed by LC-mass. At Wuxi AppTec, the hit identification of a 200K library could be finished within 6 weeks. After receiving our biomolecular targets, assay development will be first carried out, including the protein QC and testing for testing its purity and activity. If reference compounds are also provided, tests will be run to validate as well. After the assay development section, the screening will start. As many as 400 small molecules could be pulled together and incubated with the target. After incubation, targets and binders and non-binders will be separated using our 96 well-sec plates. Then the collecting plate will be transferred to our analytic group to identify the binders. Thermal Q Exactive HF is used for this job. It is a high resolution, high sensitivity, and high accuracy instrument and has done many brilliant work at Wuxi. In the whole screening section, 2 rounds of the screening will be run. After the primary screening, the binders will be picked out and run for the second time, only in smaller pools. This is just to lower the false positive rate and help identifying the true binders. After 2 rounds of the screening done, the small molecule candidates can be then cherry-picked for binding assay or other functional assay like KD test or IC50 test. So, why choosing AceMass? AceMass requires little for assay development. You don't have to know the binding site. You don't even have to have a true binder as a positive control. And as I mentioned at the very beginning, there is no bias in target small molecule binding mechanisms. As we all know, a target's biological function could be elusive. However, it won't affect AceMass screening. And since it's in solution, all binding pocket of the target is opened up for small molecule mixture. And it has more potential to identify a novel binding site. AceMass is also scalable in the case of Wuxi AceMass platform. As many as 400 compounds could be pulled together and tested at the same time. AceMass has broad applicability. It works for any soluble target and any library mixtures. So at Wuxi AppTec, we have our origin AceMass library, which is around 290K compounds. They're filtered by RUS, panes, and our structural filters. And as you can see, it shows in the slides, the library has a broad range in drug-like chemical property distribution. Okay. The AceMass platform at Wuxi AppTec is pretty mature. The target types we tested are wild spreading, including kinase, epigenetic protein, enzyme, protein complex, a protein-protein interaction target, and linked RNA. We also have a large compound library, as I just introduced. Several compound management systems could be used in our data processing to provide strong support, and strong biophysics assays and bioassay capability provide strong backup for hit confirmation. We also have great mass analysis platform, not only our high sensitivity, high resolution, and high accuracy mass instrument, we also have powerful and reliable hardware and softwares. In the end, I'd like to talk about our brilliant team. We have many experts in the team with strong scientific background and assay development capabilities. Our AceMass team is very experienced, and we have served over 28 clients, screened over 24 targets, sizing from 12K Dalton to 400K Dalton. And we also screened over 3 million compounds. And that's all for my presentation, and thank you for listening.
Thank you so much, Benny. Next up is Dr. Alex Satz. Alex has 15 years plus experience building and leading DNA-encoded libraries, Dell technology platforms, and is currently the Senior Director of Dell Strategy and Operations at Wuxi AppTec. Prior to joining Wuxi, Alex built and led the Roche Dell platform in Basel, Switzerland, and Nutley, USA, and he helped to develop the first industrial-scale Dell platform at Precris Pharmaceuticals, later purchased by GSK. Alex has also worked as a medicinal chemist at AstraZeneca, and he was an NIH postdoctoral fellow at Harvard Medical School and earned his PhD in organic chemistry from the University of California, Santa Barbara. In the field of Dell, Alex has authored over 18 peer-reviewed articles and 2 book chapters. Please, Alex, you're up next.
Hello, my name is Alex Satz, and I'm going to be talking to you about the Wuxi Dell screening platform. So let me jump right in. First, let me talk a little bit about how our Dells are actually made. So let's say we have a mixture of 3 different things. We have structures shown here and barcodes shown here, and they're connected by a flexible tether. So we have a mixture of 3 different things, and we're going to split these up into 3 different tubes. So in each one of these tubes, we have a mixture of these same 3 things. And then in each one of these new tubes, we're going to add a new synthon, which creates a new structure, which is shown here. And we're going to add another DNA tag, which extends the DNA barcode as shown here. And in this manner, we are going to create 9 new structures, and we're going to create 9 new barcodes. After this step is done, so we've had a split size of 3, we've created 9 new structures, we can pool those 9 new structures inside of a single tube, and at any point in time, we can sequence off what the barcodes are, and from the sequence of that code, we will know what the structure is that is attached to that barcode. And so this is a single split size or a single step of split and pool combi-chem. And this is how we in Dell make a lot of chemical structures in a very short amount of time and at a relatively low cost. Of course, we don't use split sizes of 3, we use split sizes of 96 or even split sizes of 9,000, and so that we can create in this manner millions of new structures that we can screen. So thus far, we have synthesized over 280 different unique Dells, which means we have created over 65 billion plus structures, which can be readily screened against a target of your choice. So how do you access these Dells and these novel structures to be able to screen them? Well, we offer you 3 different ways that this can be done. So in the first case, you send your target to us and we screen it for you. And we can screen either a collection of 150 Dells, or we can screen our covalent Dells, which has 50 plus Dells in it. An alternative to that is if you are an academic lab, we can send you a Dell Open Kit that contains 30 of these Dells for free. And you can screen your target in your own lab, and then you send the results back to us and we tell you what your hits were. Or if you are a startup, or if you don't want to send your target to us at Wuxi because you want to keep the name of your target a secret, you can order a Delight Kit, and that contains 58 different Dells. We can send those Dells to you. Again, you screen them in your own lab, and then you send the results back to us, and we then do the rest of the work and share the results with you. So how does this workflow work? Well, first, how does the Dell screen work? So the Dell screen works by taking the target of your choice and immobilizing it onto a resin or a bead. You then incubate the Dell that we provide to you with the target, and most library members will not have a ligand that can bind to your target. And so all of these library members that don't bind are washed away. And what we're left with are the members that do bind to to the protein target. And we collect these and we sequence the barcode. And from the sequence of the code, we know what the structure was that led to the binding. And then we share those structures with you, and those are the actual hits. So the workflow for this is always the same, whether you send the target to us or we send a kit to you and you run the screen in your own lab. The difference is, of course, this first step. Is the screening done by us or is the screening done by you? But regardless, the wet lab samples from that screen are then sent to us at Wuxi, and we do the rest of the work to determine what the structures of these hits were. And finally, we at Wuxi, we have screened over 184 different targets. And so the question is often asked what to expect from these screens. And of course, what kinds of targets do we find are successful in DEL screens? So I'm providing you here a list of different target classes that we have screened thus far. And shown in dark blue are the number of times that we have found a confirmed hit against that target. Shown in light blue are the programs which are ongoing right now. And shown in this light gray are the programs which have closed and which we weren't able to find a hit for. So not against every target do we actually find a confirmed hit, but against 95% of these targets we actually do. So I'd like to thank you for your time. And with that, I will hand it over back to Chris. Thank you.
Thanks for a great presentation, Alex. The following speaker is Dr. Saskia Willinger. Saskia is a biochemist by training with a great passion for biophysical techniques. She is currently the senior scientist and deputy head of the biophysics and screening department at our Krellex site in Munich, Germany. Before joining Wuxi AppTec, Saskia has over 6 years of industry experience with biophysics and analytics at Sanofi R&D in Paris, France. And before that, she obtained her doctoral degree at the Max Planck Institute for Biophysical Chemistry in Göttingen, Germany. Saskia, the floor is yours.
Hello, everyone, and thank you for the introduction. Today, I would like to draw your attention to our fragment screening and hit validation capabilities. The HITS platform is part of Wuxi's research service division. At the beginning of 2020, Wuxi's DEL screening unit joined forces with Germany-based Crystallography in Munich, a 100% subsidiary of Wuxi AppTec. As you can see, we offer services at the early stages of drug discovery programs and support our clients, especially at the target and hit finding stages. Our key expertise lies in customized protein production, DNA-encoded library screening, in short DEL, fragment-based screening, and biophysics hit validation, as well as structural biology. First of all, some general considerations about why we offer fragment-based screening. One reason is that it is complementary to classical HTS and DEL screening. In fragment screening, we work with rather small molecules with a size of 200 to 300 Dalton. Due to their size, they can bind to small pockets available on protein surfaces and thus provide a good starting point for inhibiting protein-protein interactions. One big advantage is that fragments are quite simple chemical starting points. This allows for more flexibility during hit-to-lead optimization. The fragments can be grown, linked, or merged. And this allows a very efficient way of sampling chemical space. The chemical simplicity also allows efficient optimization of pharmacokinetics in parallel with potency as the fragment hit grows into a clinical candidate. Finally, hit rates from fragment screening are excellent indicators for the druggability of a protein target. Before we dive in, a few words about the design of our fragment library that was done with strong support from medchem experts at Wuxi. The library contains fragments from e-molecules, lab network, and proprietary Wuxi fragments. To select appropriate fragments, we applied various filters to remove frequent hitters, known false positives, molecules with reactive groups, or that interfere with assay formats. All fragments are synthetically accessible, show appropriate complexity, and are Rule of 3 compliant. In summary, we selected 3,100 fragments representing key pharmacophores. Now let's talk about the starting point and a very critical point for fragment screening, the protein production. At HITS, we offer customized protein production solutions. We tailor protein towards the needs of the screening format. We offer recombinant protein production in different hosts. At Quillox, we also have very high standards for protein quality, as it has a large impact on the quality of biophysical screening and structural data. For every lot that we produce, we run SDS page and analytical sec to check for purity and monodispersity. We perform intact mass and mass spec fingerprint to validate protein identity and post-translational modifications. And last but not least, we confirm protein activity by making use of our biophysical assay platform. In summary, we always produce proteins that are the best possible solutions for downstream assays. Over decades, we have produced over 1,500 proteins. Once the protein is available, we are getting to the biophysics and screening stage. In our biophysical and biochemical assay platform, we offer more than 10 different assay formats. Currently, for fragment screening and hit validation, our main working horses are microscale thermophoresis, in short MST, the second generation instrument Dianthus that uses the phenomenon called TRIC, nano-DSF or classical DSF, and SPR. These methods offer fast turnaround times. We also have the flexibility to change between different methods depending on the target's requirements. We have optimal starting material in hand and can check for the best possible assay format. Only once we have identified the best assay format for each target, we start the screening campaign. With our platform, we can not only identify hits, but characterize them in detail. We can determine binding affinity, kinetics, and thermodynamics. Besides our working horses, we also use NMR, ITC, DLS, ASMS, FP, or biophysical assays as routine assay formats. Now I want to give you a more detailed overview of our screening workflow. First, we produce the protein and run a QC by SDS page, analytical sec, and MS. Then we use nano-DSF to optimize the assay conditions. We screen for an optimal assay buffer, detergent, and DMSO concentration. We also confirm protein activity using tool compounds. Based on this information, we perform an extended assay setup. We investigate which technique, MST, Dianthus, or SPR, is most suitable for the screening. Once the technique identified, we run a single point screening followed by an affinity ranking of the identified hits. And this typically takes 2 to 4 weeks. Afterwards, the identified hits are validated by an orthogonal biophysical or biochemical assay. We also recommend our clients to set up a suitable crystallization system in parallel. This can be a soakable or a co-crystallization system. Further down the road, we can support you by SAR by catalog or with in silico approaches. In the following 2 slides, I will show you some real-life data of a case study that we performed with the stimulator of immune response genes, in short, STING. The construct was designed with a His and an AppTec, such that fragment binding could easily be analyzed by MST and SPR. For MST, we established non-covalent labeling of the His sec with red tris NTA dye. The assay was validated by demonstrating reproducible binding of 2,3-CGAMP, the natural substrate of STING, with 10 nanomolar affinity in line with published data. We ran a primary screening at 1 concentration, followed by a KD affinity ranking. And the best fragment hits exhibited binding with KD values in the 2 to 3 digit micromolar range. For orthogonal validation, we made use of our assays that we established in-house. On one hand, by SPR, immobilizing STING to a streptavidin chip by the bioattenuated AppTec. On the other hand, nano-DSF. And by that, we were able to confirm 66 binders. We also decided to validate a subset of these binders by STD NMR, and the data was really encouraging. 9 out of 11 selected hits were confirmed by NMR. After biophysical screening and hit validation, typically a next step in these kind of projects is structural validation of the hits by X-ray crystallography, which we also offer at HITS. So you will find here a general overview of the crystallization workflow. Ideally, we establish a soaking system for the target protein. This enables setting up crystallization trials at very high compound concentrations. The data is automatically processed and hits are manually refined. In case of success, This opens up all possibilities of structure-guided drug design and would then also involve our MapChem experts at Wuxi. With this, I would like to conclude my talk. I thank you for your attention and I'm happy to answer your questions.
Thank you, Saskia. That was great. Our next and final speaker is Dr. Dave Match, who will talk about virtual screening. Dave is responsible for the preclinical discovery platform with which Wuxi AppTec supports its collaborators in Europe and Israel. With a background in medicinal chemistry, Dave joined Wuxi AppTec 7 years ago. Before that, he worked in biotech as the VP of Research at Santion. He founded NCE Discovery, now DomainX, and supported an integrated biomedical research group at the University College of London. Dave started his career at the Wellcome Foundation and received his PhD in chemistry from Imperial College London. Please note that after this presentation, our speakers will remain online for a panel discussion with Q&A opportunities. Dave, the floor is yours.
Hi, everyone. Thanks for staying through to the last section of this presentation. My name's Dave Madge, and I represent our discovery business in Europe. Today, I'm going to talk a little about the current status of virtual screening as a hit finding strategy in our organization. As you will know, virtual screening is a collection of computational techniques we can use to mine very large libraries of compounds to prioritize them for further evaluation. This might include further computational evaluation, acquisition, or synthesis. One important aspect of this technology is, of course, that the molecules we screen using these approaches do not need to exist as real samples. In fact, they may never have been synthesized. So we can apply a computational selection tool to imaginary molecules in order to decide which ones might tell us something interesting about interactions with the target protein and may therefore be worth synthesizing. Such virtual libraries can be very large, many billions of molecules, and can also be very diverse, encompassing large areas of chemical space. We can apply these technologies based on our understanding of one or more ligands with the target protein, the relation of a collection of known ligands to each other, or sometimes just based on our knowledge of the three-dimensional structure of the target protein. Of course, we will usually want to follow up on a virtual screen by carrying out a real measurement of the biological activity of the molecule. Hence, there's been a lot of interest in the creation of so-called make-on-demand libraries. These are virtual libraries based on the enumeration of known chemical reactions using available scaffolds and building blocks. And consequently, there is a realistic expectation of fast follow-up, making real samples available with good success rates. One way to think about this is that virtual libraries store a huge amount of nascent information on small molecule protein interactions, information that can be revealed using virtual screening platforms. So as the computational tools that enable screening of large virtual libraries have improved, enabling screening of larger libraries in shorter time periods, so have the size of the virtual libraries grown, limited only by our creativity, and in particular for make-on-demand libraries, the availability of building blocks to include in virtual library enumeration, whilst retaining reasonable access to the real molecule. High-throughput searching paradigms for such large virtual libraries are becoming very established. And this figure refers to work carried out by John Irwin and colleagues, docking around 100 million make-on-demand small molecules against the AMP-C beta-lactamase and 140 million against the dopamine D4 receptor. For both targets, new high-potency chemotypes were identified from the huge chemical space being screened, with nanomolar and even picomolar actives confirmed in biochemical assays. These results were backed up with real X-ray data, which showed a strong correlation between the predicted binding mode and that confirmed by crystallization across multiple ligand types. In parallel to the year-on-year increases in raw computational power we've all come to expect, there are increasing applications of machine learning to support an even faster application of virtual screening techniques. By applying machine learning, we can rapidly build predictive models based on smaller data sets, which we can then apply to much larger data sets rapidly and with a high degree of consistency and accuracy. This means we can screen ever larger virtual libraries in the same time or less scanning chemical space encoded by billions of structures. A recent example of the transformative impact this can have is shown on this slide, from a study using machine learning to model the relationship between structure and antibiotic activity. A neural network was trained on about 2,000 known molecules, whose activity data was collected and then classified simply into active or inactive, and used to train a deep neural network. The model was augmented with molecular features computed by the RD-Kit tools, resulting in a receiver operating characteristic curve, or ROC, area under the curve of about 0.9, demonstrating the highly predictive nature of the model. The model successfully identified and classified further known antibiotics from small data sets, and was also used to screen 100 million small molecules from the zinc dataset, identifying novel molecules with confirmed in vitro antibiotic activity. To reinforce this trajectory of larger libraries and faster docking performance, a recent paper describes the use of a deep docking approach. This work was based on a deep neural network utilizing QSAR models trained on actual docking scores of a small subset of a compound database, and then predicting the docking scores for the rest. This circumvents the need for vast computational power and makes screening of billions of molecules feasible from both a time and a computational power perspective. High ROC values were observed across multiple targets, demonstrating the scope and usefulness of this strategy. It's often suggested that there may be 10 to the 63 possible drug-like molecules. And so, of course, we cannot construct a virtual library to represent all of those compounds. Hence, we need to perceive ways to explore that space from a much more focused starting point. One strategy that is attracting increased attention is to combine generative machine learning models able to carry out in silico design of new molecules based on a set of chemistry rules, with virtual screening based on an evaluation of fit to a structural pharmacophore. We can then envisage an iterative in silico design and docking cycle using feedback and retraining of the generative model to help us explore chemical space that the algorithm creates. In doing so, we can also incorporate additional filters that use predictors of other metrics, such as basic physical chemical properties, DMPK properties, and toxicity prediction. This machine learning-led strategy gives us a new tool to discover novel molecules that were never part of any real or virtual collection. This slide provides a slightly more detailed description of this workflow, which can start from virtual or real hits and uses a combination of different representations of the active molecules, such as molecular fingerprints and feature vectors, to train a deep neural network and enable this multifactorial property prediction and molecular optimization approach, driving generation of new molecules we could not have conceived of by other routes. So to close this section of our discussion, virtual screening forms one part of our comprehensive toolbox for identifying and optimizing chemical matter for new targets. We can envisage matching each platform to the target of interest based on the target type, tractability for different platforms, and pre-existing knowledge. But it's also worth considering how we can combine the application of more than one platform and create a much more diverse and comprehensive collection of starting points for drug discovery by evaluating more chemical space, including generating completely new chemical space for our targets. Many thanks for your attention.
Thank you, Dave. With this, we have had 5 expert talks about the different screening approaches which could help and accelerate hit finding and expansion endeavors for your projects. It's important to us to help you find the best chemistry and the right hit finding approach. By leveraging multiple technologies, rapid and successful hit finding is enabled, delivering novel, tractable chemistry for your targets. Together with our clients and partners, we have run hundreds of successful hit finding campaigns. These are fully integrated with medicinal chemistry and in vitro biology and ADME for lead optimization. It's now time for a Q&A session with our experts who will answer all your questions.